newspapers.
Good afternoon, students. Good afternoon.
Good afternoon, ma'am.
So,
Good afternoon, ma'am.
how many of you have joined?
49.
49.
39 members, ma'am.
Yeah, that's a good number. We'll try to start. I'll just come in a minute. Okay, before I start my video, what is that we did? Any one of you all, can you all tell me what all we studied yesterday? Any one of you all? What was the topic we completed yesterday?
Life cycle of Elphidium, ma'am.
Life cycle of Elphidium. Before that?
Protozoa.
Protozoans, yes. So what are the features of protozoans? What are the features of a protozoan? First feature, are they multicellular or unicellular?
Unicellular.
Unicellular. And what more characters you found in them? So didn't go through, I suppose. Is your book open? All of you all are having the books. Before the textbooks, I want you all to have a rough book with you all, with a pen, so that any important points you can just write it down, because everything you cannot search in the PowerPoint presentations that I'm going to give you all. So for an examination point of view, if you just note down some important points, that will be very useful when you all are going to an exam. So it gives you all a little bit of gist of what all you studied previously. To go through all the PowerPoints is difficult. Okay, did you all get my point? So just make a note of some important points, and then that should be good enough for you all for your examination point of view. Okay. Okay, then coming to today's class, we will. So till yesterday, what was the last topic we did? Yesterday's topic, what was the last one we did? No idea. Some of you all were absent.
Reproduction and cycle, ma'am. Polystomela. Polystomela.
Very good. Polystomela. So polystomela means we were talking about type study of. What was the type study we did in polystomela? What was the other name of polystomela? To be very precise.
Elphidium. Elphidium.
Elphidium. Exactly. So we were talking about Elphidium. And on the Elphidium, we were talking about two different kind of forms, that is. Yes, we were talking about two different forms. What are the forms? What were the different forms that we were talking about?
Megalospheric and microspheric.
Okay. What is it? Yeah, what is this now? Megalospheric, megalospheric form and microspheric form. So what is the difference between a megalospheric and a microspheric forms?
Microspheric means asexual reproduction. Megalospheric means sexual reproduction.
Okay, and what are the other forms? What are the other differences that you find in them? Proloculum. Just check the proloculum. Yeah, proloculum is large in megalospheric and small in microspheric.
And
right?
uninucleate.
Uninucleated. Uninucleated in which form?
Megalospheric.
Megalospheric. Very good. And we saw even the alteration of generation between the megalospheric and the microspheric form. So now coming to the next topic, we will be talking about some protozoan diseases. So pertaining to this class, we are going to talk about some protozoan diseases. So before just going into the protozoan diseases, I would like you all to just check these classifications again. We were talking about subphylum. We had four subphylums: Sarcomastigophora, Sporozoa, Nidospora, and Celiospora. Under Sarcomastigophora superclass, we talked about Mastigophora. Under these, you have four classes. All the blue color ones are the classes. Under these four classes, we were talking about Phytomastigophora, the Zoomastigophoras. Then you have superclass Opalinata, pink colors, and Sarcodina. Under Sarcodina, we are talking about two different classes, the Rhizopodia and what are the other ones? We were talking about the Pyroplasma and the Actinopodia. And under Sporozoa, we were talking about Telospora, Taxoplasma, Haplosporia, Nidospora, Myxosporidae and Microsporidae, and Coelophora is ciliata. Now coming to the protozoan diseases. So as I told you all, the protozoan diseases, you have different types of diseases due to protozoans. Some are they are very parasitic in nature, some are mutualistic in nature, and some are not at all parasites. That is what we were talking about, right? So these are a few examples that I felt would be informative for you all to understand. For example, if you're going to talk about the malaria disease, what are the organisms that is going to have these problems, creating these problems? So you have this Plasmodium falciparum, vivax, malaria, and ovale. Among all these species, falciparum, Plasmodium falciparum was found to be very, very highly parasitic and dangerous one, because that parasite leads to cerebral malaria. Have you heard of the cerebral malaria? What exactly you mean by cerebral malaria? Anybody? What exactly you mean by cerebral malaria? No idea? Cerebral means brain. The malaria that affects the brain is called cerebral malaria. Mostly in the African countries, you come across this particular problem. As I said, this is very parasitic and very pathogenic in nature, and they can be even fatal. When you say fatal, that means the person is definitely going to die. And once the brain is affected with this malaria, then definitely the patient cannot survive for a longer period of time. So that's what is falciparum is all about. And they ask us to be very careful, because we are unable to control this malarial parasites, because these protozoans, the malarial protozoans, they are so intelligent that even though we have so many, we try to have this medicines, DDTs, vaccines, and all this, we are unable to still control this malaria, because the falciparum and the vivax itself, the DNA is so well manageable that they keep on changing their protein, the outer covering of the protein, what you call. For example, we were talking about this COVID all these days, the COVIDs, wherein all this vaccination was produced so that they can study the protein, the surface protein, they find out a particular antibody that can go and affect, join the antigens, and then they are going to be destroyed. But here, this particular protein of the DNA, they are going to keep on changing. So a particular antibody, we are unable to find out to control them and kill them. So that is the basic importance of here in falciparum. There are so many research going on. And as students, what I wish is uh, just degree is not enough. What we have to do, we have to think about whatever we are studying, you have to put it into a practical use in the future so that you can also come up with some new therapies or new medicines, because as a zoology students, we are not just here only to read whatever is there in the textbook or your knowledge is being given. You have to improve your knowledge by going further into research. Research comes only after completing your PGs. Fine. If you have any interest in doing a PhD, you can also ask me the questions regarding how you have to go about, what are the topics you have to take to complete your PhD, so that you can become professors in the future. Fine. And this is just a list I've given to you all. You can just go through all this list for your information point of view, because this type of questions you might be asked in your MCQ questions for your further, if you have going to attend any examinations. You just go through this. This will be of some help. Coming to the first one, we will be talking about Leishmania. So here under Leishmania, this comes under phylum Protozoa, superclass Mastigophora, genus Leishmania donovani. So this is the picture that you're going to talk about over here. Remember these two pictures over here. This is pro-mastigote stage. The elliptical elongated structure is a pro-mastigote stage, whereas this spore-like structure is an amastigote stage. So this is the infectious stage over here, amastigote stage, or also cyst stage, what you call them. So you can just remember pro-mastigote stage and an amastigote stage. So what is exactly happening in this particular one? So these are vector-borne diseases. So when you call them as vector-borne diseases, they need some vector to carry them into the infectious stages. And these are caused by what fly? What fly is this caused by? This is caused by sand fly. Can you see this picture over here? So this is a sand fly over here. And this is the Leishmania pictures of there, the elliptical ones. And you can also find some encysted ones over here in between. So this is a picture, what you can check over here. And some are undergoing binary fissions over here. You can see there are two. This is a single one, and this is a binary fission undergoing, that is to produce and multiply in a number of parasites over there. So these are digenetic parasites which require two hosts. Di means two. They require two hosts. One is a primary host and one is a secondary host. Primary hosts are usually the humans, because whatever diseases that we are talking about will be definitely on the human beings. So primary means first. The first is the humans, and the secondary host is where they have to lodge themselves so that they can survive, they can reproduce, they can grow in number, and then again reinfect the humans. So primary and the secondary host. So they are vector-borne diseases. And in a primary host, the parasite feeds and multiplies itself asexually, whereas in the secondary host, they multiply themselves asexually. So that is what the primary and secondary hosts are mainly useful for. And these are usually in an invertebrate or blood-sucking insects. So only these sandflies, which are blood-sucking ones, can ingest all these parasites, and they are able to send it back into the human beings. And the sandfly belongs to genus Phlebotomus. So that's a blood-sucking fly. What's the name of this fly? Sandfly. So this is the picture that I want you all to again understand. Pro-mastigote stage and the amastigote stage of Leishmania. So what exactly is happening over here? People, when infected, they are bitten by an infected female sandfly. And the sandfly injects a form of protozoa called promastigotes. Now remember, promastigotes. What is the shape of a promastigotes? This is an elongated one. So this is the elongated structure. So people are infected when they are bitten by an infected female sandfly. Infected female sandflies, which is already having this particular parasites present in them. So when they are biting a human being, they inject a form of protozoa called promastigotes that can cause infection. Once these are ingested inside, these promastigotes are ingested by a certain immune cells called macrophage. What do you mean by a macrophage in the circulation? Anybody? Macrophages in the circulation. Example, I'm giving you all some information also. No idea? These are all the WBCs. Clear? Am I audible, students?
Yes, ma'am.
Yeah, I didn't get any response. I was just wondering whether I'm audible or not. Okay, fine. Macrophages. Macrophages are nothing but the WBCs that are circulating in our blood system. So, what is the function of a WBC? RBCs and WBCs you have, right? So, what is the function of a WBC? It's an immune response, phagocytosis. Whatever the microorganisms enter in our body, they are being phagocytosed. They act as police in our circulatory system, wherein they catch hold the thieves and the robbers. In the same way, our WBCs are there to protect us from all these microorganisms. So what happens is this promastigotes, they directly go ingest is they enter inside, ingest by a certain immune cells called macrophages. What are macrophages? Again, I'm telling you, white blood cells are also called as WBCs. In these cells, the promastigotes develop into another form. What's the other form? Amastigotes. And how does an amastigote look like? It's a cyst form, the round structure that I showed you all. So this is an amastigote. From this shape, they change themselves into this shape in WBCs. Once the amastigote enters the cells of reticuloendothelial system. What are the reticuloendothelial system? They can be a spleen, they can be in the bone marrow, they can be a liver, and they can also be a lymph nodes. What do you mean by a lymph node? Lymphatic cells. These are also immune in nature. That is, they are phagocytic in nature. That is, they help us from all different types of microorganisms. And where all you find this lymph nodes? Any idea where all you find this lymph nodes? You find them at the neck region over here in the neck region. You also have those lymph nodes in the armpits. Then there's also a Peyer's patch, what you call. You find them in the stomach region and the intestinal region. And the lymph nodes are also found behind the knees, on the legs, behind the knees. You have this lymph nodes. They are something like grape-like structures, small grape-like structures, lymph nodes. And these are the one that filters, the lymphatic tissues are being filtered over here, and any microorganisms that are moving in the lymphatic tissues, they capture them, they phagocytose them, and they throw them out of the system. So why usually in the neck region, in the armpit region? That is, what we do usually is we come across there's so many different microorganisms in the air, right? We are breathing air. So once when you're breathing air, it can go inside. And once they are going inside, they have to be cleaned up. And the first line of control is the lymph nodes that are present in the neck region. And if they try to escape that neck region, there is one near the armpits. So at each and every level, your body is trying to purify your blood so that it is trying to remove the microorganisms present in the circulatory system. And that is the reason why we are able to still survive, even if the environment condition is not so good nowadays, with so much of impurities that are present over there. And not to forget, I just love to keep telling my students about this pani puri, right? We all are lovers of pani puri, and I don't know from where they get that water, what type of materials they use in that, and which type of hand they remove and give it to us, and we happily take a gulp of it. It might be having so many microorganisms, and still we are able to survive with all these microorganisms. Why? Because we are having this WBCs and the lymphatic cells, which is trying to protect us from different types of microorganisms. Okay, coming to our class over here. Now that you have understood what a lymph node is, and there is when they enter all these type of reticuloendothelial systems, they undergo slow multiplication by binary fission. I showed you all in that picture how they are undergoing binary fission, and because of this different types of organs significantly enlarge. That is, when a microorganism is increasing in size by binary fission, definitely they will become enlarged because of the infections that is present inside. Because anything foreign material is, your body is not going to accept it. Even it can be even a minute microscopic one, it is still a microorganism for our body, and it's a foreign material for our body, and definitely it will not be accepted by our body. And they try to keep fighting, but if your immune system is compromised, that is, if you don't have a proper immune system, then definitely this is going to increase in size in your body, because the immune system is not able to remove them out of a body. So when the number of parasite reaches 50 to 200 or more, the macrophages gets ruptured, because macrophages are very tiny ones, and imagine 200 parasites present in that small RBC or WBC, they are definitely going to get ruptured. And once they are ruptured, they releases the parasites. And when the sandfly bites an infected person, all these are being taken up, and again they are going back into that reproductive cycle, where it goes and infects another human beings. So the infected, when a sandfly bites an infected person of an animal, it becomes infected by ingesting the blood containing macrophages with a mastigotes inside them. So what happens over here? You can just have a look at this picture over here. So what is happening? First thing, just take this first example over here. The sandfly takes a blood meal in which is present with pro-mastigote stage in the into the skin of the fly, and when they are taking the blood meal, this injects the pro-mastigote inside the human beings, and once inside the human beings, the pro-mastigotes are phagocytosed by the macrophages or other types of mononuclear phagocytes. Here, if your immune system is perfect, you're able to phagocytose them. You're able to phagocytose them. If your immune system is not good, then this pro-mastigotes enters into the WBCs, and they change into amastigotes. You can see those tiny, tiny ones in the WBCs. This will grow in size. They develop by binary fission, and all these are, once the WBC is reaching 200 cells inside, they rupture, and all these amastigotes goes and again infects different parts of the body, trying to increase all those reticuloendothelial cells in size. And meanwhile, when a sandfly takes that same blood, the blood is taken out, and all the promastigotes, they enter the fly, and then they are transforming into amastigotes, ready to be infected into another human being who is not having all these parasites. So you have to be very careful with different types of this disease. So what exactly is this outcome of this particular disease? So outcome is, this is also called as a dum-dum fever or a Kala Azar fever. Kala Azar is usually known to us. A dum-dum fever is very rarely known, but this is also called as a dum-dum fever, which is a serious oriental disease of man caused by leishmania, also called as Kala Azar. What is Kala Azar? According to Indian word, Kala means black, Azar is a disease. A black disease is known as Kala Azar disease. And this is prevalent mostly in India, China, Mediterranean countries, and of course some parts of Africa and South America. Early symptoms includes swelling, high fever, enlargement of the organs or reticuloendothelial systems. Reticuloendothelial system means all the liver, the lymph system, and all those other endothelial system, which gets the spleen, which gets enlarged in size. followed by general weakness, anemia, definitely, because all your RBCs and the WBCs are being used up by these parasites. So definitely you will have anemia. What do you mean by anemia? Loss of blood is anemia. And as they are being reduced, the number of cells are darkening. That is, your RBCs becomes very dark because of these parasites. Hence, your skin becomes dark in color. That is where it is called as Kala Azar. And in advanced stages, advanced is completely wherein there is no other bringing back of the person over here. The last stage, what you call them as the advanced stage, the skin of the infected person becomes completely dry, rough, and dark with lot of pigmentation. Pigmentation is when the body becomes dark in color. That is what you call as pigmentation. And they become brittle and fall out. And if the patient does not receive proper treatment, the patient can die within 2 years. So that dangerous is this Leishmania donovani, which is causing Kala Azar. And this is a picture of a patient suffering from Kala Azar. You can see the enlarged spleen and the enlarged liver over there, because mostly you don't find such type of a belly in a small child. But this enlargement is because of the development of the leishmania over here, they increase in size. And also you can see the pigmentation on the skin. So the dark spots that are present, these are the ones that are caused by this Kala Azar. So this is the first disease. And coming to the next one, we'll be talking about Entamoeba histolytica superclass Sarcodina. So what do you mean by a Sarcodina? I told you all. Anyone? Sarcodina means fleshy. Write down over there in your notebook. Sarcodina means flesh like parasites. That is, they are very soft and fleshy in structure. So that is what is called as Sarcodina. What do you mean by rhizopodia? Rhizopoda, rhizo, root, root like foot. Poda means
Root foot.
yes. Rhizo means root, poda means foot. Root foot is the meaning of rhizopoda, and genus Entamoeba histolytica. So this is the picture of a trophozoite Entamoeba histolytica with a pseudopodia over there. You can find a pseudopodia or false foot, the nucleus present over there, the foot vacuoles, and the ingested RBCs. So we are going to talk about the Entamoeba histolytica disease over here. Life cycle of Entamoeba histolytica begins in a single host, hence in a single host. That is, they don't need a vector. They don't need a secondary host. A primary host is enough, and the primary host is none other than human beings. So whatever we are talking in science is all about human beings. We want to have a better life. So that is the reason why we study a lot about humans and human diseases. Okay, there are some animal also where veterinarians do study, but because we are mostly interested in the human diseases, so we'll be talking mostly on humans. So this histolytica needs only one host, that is individual human host. And the disease caused by this histolytica is also called as amoebiasis or amoebic dysentery. So anyone who are infected with this amoebiasis, they have this food poisoning. Have you ever come across the name food poisoning? And whenever you have a food poisoning, you definitely have dysentery. Okay, so that is amoebiasis. And sometimes we need to be very careful. There's a lot of antibiotics to be taken when we are affected with amoebiasis. So we have to be very careful when we are having any food outside. I'll just tell you one more important tip. Like whenever we go to any marriages or any functions, we are definitely being treated with salads. Okay, so something like onions and carrots and all those salads over there. Specifically, try not to touch onion salads. Why? Because the onions, when once you cut it open, within half an hour, you will find thousands to lakhs of microorganisms growing on top of the onions. So try not to take onions, even at home also. If you cut open an onion, Sometimes we need only half part of the onion, and the rest of the half we just leave it aside or keep it inside the fridge so that you will want to use it later on. Never do that. You either throw that onion or take a small onion whenever you want to cut a small ones, but never try to eat a onion that is open for a longer time. This applies same where when you're going to eat pani puri, don't take onions, because we don't know when he has cut those the onions and kept it. It definitely can be more than half an hour, and there are so much of microorganisms growing on those onions. Okay, so that leads to this amoebic dysenteries also. So coming to our topic over here, life cycle of Entamoeba histolytica. Infection. This is the first stage. The first stage is the cyst stage. Cyst. Now you know what a cyst is. The spore-like structure is a cyst. Infection occurs by ingestion of mature cysts through fecal contaminated water or food. Fecal contaminated water or food. So what happens over here? We must have heard sometimes in some places where the water has been contaminated by the drainage water. So, mostly that happens. You come across with complaining to the water board where the mineral, water has been contaminated. This happens usually in the rainy season. Yes. So when the water is being contaminated is when the drainage water gets mixed up with the drinking water, and when you try to drink this type of water, we are ingested with this cyst. Because thankfully now, due to Swachh Bharat, we don't see most of the people defecating in the open, right? That is the main reason where the water gets contaminated with the fecal matters, right? So try to have lots of Swachh Bharat going on in your areas. Also try stopping others from defecating in the open uh, areas. So due to protection from the walls, the cysts survive for several days and sometimes for weeks, and they are responsible for transmission. And excitation. Excitation is the cyst is being opened out, is the second stage. When the cyst enters the ileum of the small intestine of the host, the process of encystation begins. What is the encystation? Removal of the cyst. And the process by which the cyst transforms into trophozoites. Now, this is the main one which infects the intestine. The trophozoites are released into the small intestine, and from where they migrate to the large intestine, and they try to survive over there. So this is a picture of a trophozoite. So this is the apical complex where they make a hole into the fleshy areas of the ileum, and then they try to take the food from the ileum and thereby infecting us. So this is a trophozoite stage, and this is a cyst stage. The first stage of the cyst, that is a round structure, is a cyst stage. Now, the trophozoite stage over here, trophozoites are unicellular parasites that measures up to 14 to 18 micrometer in diameter. So they are that tiny, multiply in the small intestine by binary fission to produce cyst, and they exit via human stool. So until they are trophozoites, they are still present in the small intestine. And once they become cyst, they exit via the human stool, and several trophozoite remains into the lumen of the small intestine, and they keep trying to destroy the internal mucosal lining of the intestine. Internal mucosa. What do you mean by mucosa? The mucus secreting cells. Okay? In the intestine, you have the mucus secreting cells. That is what it's called as mucosa. Why do you need mucus secreting cells inside the cell wall of the intestines? Any idea? Do we have mucosa in the stomach walls? Anybody?
Yes, ma'am.
Yes. Why do we have the mucosa in the stomach wall, ma?
For
That's
digestion.
protection against the concentrated acids released during digestion.
Very good. Very good. That's a good answer over there, because your stomach keeps on releasing hydrochloric acid because you have to digest whatever food you have to take in. The hydrochloric acid is so dangerous. Have you ever witnessed a drop of hydrochloric acid falling on your skin? It burns your skin. So when it can be so concentrated, what will happen into a stomach? Because definitely you will have almost around 5 ml to 10 ml of hydrochloric acid in our stomach. It is as good as drinking half a glass of water, right? And that will definitely spoil the cell wall of the stomach. But thankfully, we are not affected with this hydrochloric acid because they have the mucosal cell lining. They keep on secreting the mucosa, which protects our cell wall. The same thing is occurring in the intestinal mucosa. Why? Because they also have those digestive juices which are entering inside to digest the food that is present even in the intestine. And from there they enter the bloodstream, and further they grow into the extra intestinal region of the host, like lungs, liver, and brain. This is when it is very advanced stage, but the first stage is the human small intestine. And histolytica is a pathogenic infection, can be asymptomatic, means there is no symptoms at all. But if there is symptoms, then you will have definitely the amoebic dysentery or amoebic liver abscesses. Liver abscesses, the liver cells are being destroyed over there. And symptoms can include watery dysentery, blood diarrhea. So you find blood in your diarrhea, weight loss, fatigue, abdominal pain, and amoeboma. Amoeboma is tumor-like mass. That is because of the multiplying of histolytica. They form a mass or tumor-like structure around the intestinal wall. That is what is called as amoebona. And amoeba can bore into the intestinal wall, causing lesions. How are they able to bore? Because of the apical complex that I told you all in the trophozoite stage. So this is the apical complex, and this will help them to bore inside the cell wall and leading to lesions in the intestine. And by that, they may also reach the bloodstream. And a common outcome of this invasion in the tissue is liver abscesses, which can be fatal if untreated. Liver abscesses, liver cells are being degraded or destroyed. So what is the function of a liver in a human being? Why is it fatal if the liver is being affected?
The detoxification mechanism might be affected.
Yes, detoxification. And any other answer for that? What is the function of a liver?
Digestion, bile juices.
Bile juice, yeah, secretes bile juice, and also
Secretes
different
bile
types.
juices.
They secrete bile juice, and also they secrete. Fine. Please mute it, ma. Please mute your computer over there. Right. And they also secrete some digestive juices also. Yes, they have to secrete the digestive juices. Oh my God. So, Sri Vaishnavi, can you please mute chesinama? Sound chala akara? Yeah, that's good. Right. And what is happening over here, like in the stomach? Is the food being completely digested in the stomach?
No, ma'am.
No, they are not. It is only half of it. Only 50% of the food is digested in the stomach. The rest of the digestion takes place in the small intestine. And how is a small intestine being able to digest the half digested food? Is because of the secretions that is being secreted into the intestine from the liver. The secretions are secreted into the small intestine, is when we are able to digest the food. So here, when this parasites are trying to destroy the liver, then definitely we are unable to do our common work over there, and hence it can be proved fatal if untreated. What do you mean by untreated? If you're not going to take proper medications, then definitely it becomes fatal. Okay, so this is the picture of for you all to understand, that is a mature cyst enters inside and then they affect the small intestine, the trophozoites over here, and then some of them migrate outside, that is through fecal matter. They go outside and then they again come into the contaminated food into the system of a human beings when a person ingests the contaminated food. So this is the life cycle of the complete Entamoeba histolytica. Okay, coming to the next one, Plasmodium. Plasmodium, the one that causes malaria. We are unable to stop this malaria still. Malaria was known to us from time immemorial, and there are some great personalities who died of malaria. One great personality, anyone can answer who is the first person who died of malaria? Not first person exactly, but a great person who has died of malaria. Any idea? We all know that malaria is being spread by
Mosquitoes.
which mosquito?
Mosquitoes.
Which mosquito?
Female Anopheles.
Mosquito.
Female Anopheles mosquito. Yes. So when this female Anopheles mosquito is lodging all this plasmodium, definitely when it is biting us, it will definitely ingest all this plasmodium. So the great personality is Alexander the Great, who died of this malaria because he was unable to treat himself. And ultimately, though he was so great, Alexander the Great, I hope everyone knows who he is, the great conqueror, right? No one was able to withstand his strength and valor in fights, but he had to die because of this tiny protozoan called Plasmodium. So this dangerous is Plasmodium. As I told you all again, Plasmodium is very, very, very intelligent protozoan. The nucleus over here, you can see the nucleus over here. This DNA is the one that keeps on changing its protein structure, and that is the reason why the scientists are unable to come up with a proper vaccine to control this particular malaria. And how do you control malaria? Only by using specific like the mosquito coils or the mosquito nets. Only these are helping us to fight against this malarial parasites. And one more thing I want you all to see is this apical cap over here. So this is an apical cap which helps in boring into the cells of any host. So that is one particular thing I wanted you all to see. Okay, coming to the life cycle of Plasmodium. When a female Anopheles mosquito wants to suck a blood, why we call only female Anopheles mosquito and why not a male Anopheles mosquito?
Because male anopheles mosquitoes do not bite.
They
Because
don't
they
bite?
not smells plant.
Come again, come again. Your sound was not audible. Okay, let me explain to you all.
They require blood for reproduction.
Reproduction. Another better way to tell.
They
Have to
Proteins,
nourish
ma'am.
they have to nourish their eggs, right?
Yeah.
So when we are talking about female Anopheles mosquito and blood sucking, some people will think these females are always blood suckers, right? No, they are not exactly blood suckers. The mosquitoes doesn't need blood to survive. The difference is the male Anopheles mosquito, all the male mosquitoes, they can survive on fruit, the flower nectars or the juices that they get from the flowers. The females also can survive by taking in the nectar of the flowers. Only during development, that is only during reproduction, once the eggs are being formed, the zygotes are being formed, the mother Anopheles mosquito wants to nourish the eggs. So to nourish the eggs, it needs blood. Only then they come to bite a human to take in the blood. Okay, not just humans, it can even bite any living organisms, whichever is warm-blooded. So that is how they sense. Wherever the human beings are there, you send some electromagnetic sensation of that warm blood. So that is how the mosquitoes are getting attracted to the humans because they find that warmth in them and they immediately come and bite so that they suck the blood. So once they are sucking the blood, what is happening over here? All those sporozoites that are lodged in the proboscis is released inside. Why? When they have to take in blood, Why should they release the parasites in the blood capillaries of the humans? That is, when the proboscis enters inside the skin, or when they're piercing inside, they have to
Blood
release
coagulates.
an anticoagulant, because they are going to suck the blood.
Ma'am, your voice is not clear, ma'am.
What's happening with my voice? Okay, is it fine?
Okay.
Yeah. So what is happening over here is, yeah, when the proboscis is being inserted into the blood capillaries inside the human blood, what happens is, when they are sucking the blood, the anticoagulants, the coagulants will try to coagulate the blood, and the proboscis is not able to suck out the blood. So what they do is, they release some anticoagulants inside. And once the anticoagulants are released, they're also releasing the sporozoids, which are present in the proboscis. And once the proboscis are released into the circulation, they go in search of the hepatocyte. What's the hepatocyte? It's a liver cells. They go in search of a liver cells, and they try to grow over there. That is, they try to develop themselves by taking in the proteins that are present, the nutrients that are present in the hepatocytes. As they're growing, they grow in number and they become schizons. Once they have taken up all the nutrients from the liver, then there is no more food that is present over here. So what happens in 5 to 6 days, from the day it has bitten to 5 to 6 days. Normally, for the parasite to show its symptoms, it at least needs 10 days. From the day it has bitten till the 10th day, you will not know that you are being infected by this parasite, that is malarial parasite. So what is happening? First, they have to search the liver cells. Then they infect the liver cells. They try to grow in size and they become schizons. Once there are no more proteins in that liver cells, they try to burst open themselves, releasing all the merozoites in them. So all the schizons, once they burst open, they are released as merozoites. Sujata, muted or no? What is the background sound that is? Yeah, fine. Okay. The merozoites, once they are released in the circulation, they have to first search the RBCs. Without the RBCs, they are not going to survive. So the merozoites are going to first search the RBCs over here. And once they enter the RBCs, they are going to grow inside the RBCs, forming into a next stage called the trophozoite stage. A trophozoite stage is nothing but a ring-like structures. Can you see this ring-like structure? So this structure is called a trophozoite stage. Now this trophozoite has to develop and increase in number. And how do they do that? They try to take away the proteins that are present in the RBCs. And what is a protein that is present in an RBC? Hemoglobin. They're going to take up the hemoglobin and they're going to change all the hemoglobin into hemozoin. Hemoglobin is changed into hemozoin, Z-O-I-N, and that hemozoin is toxic in nature to us. Hemoglobin, when converted to hemozoin, that becomes toxic to us. So once the trophozoites have increased in size, they become schizons. And once the protein part is completely taken up by the growing parasites present in the RBCs, they are going to burst open. And once they are burst open, all these merozoites are released into the circulation once again. And when this RBC is broken, now this is a dead RBCs over here. And what happens is the hemozoin that was present inside this RBC is also ruptured and they also go into the circulation. Is when we are affected by these parasites. What is happening? Once the hemozoin is present in the circulation, you must have seen in the malarial patient, they sometimes suddenly they start shivering, right? Have you seen that shivering? They shiver a lot, and the temperature also rises to 101 to sometimes even 1 or 2 degrees centigrade. Okay, so why does that happen? is because of the hemozoin present inside the ruptured RBC is released into the circulation, and that hemozoin is toxic in nature for the human circulatory system, and is when you have this high fever and body chill, no matter how many blankets you cover that patient, the patient is completely shivering with tremors. That's because of this hemozoin content that is released in the circulation. Now, this merozoites, once they are out of the RBCs, they will go and search for another fresh RBCs, they infect, and the life cycle goes on until you are taking some drugs to control this particular merozoites from growing. When I'm scientifically, when I'm talking about drugs, I'm not talking about cocaine or something like that. Drug means medicine. Scientifically, drug means medicines. Okay. Now, some of the trophozoites over here, due to unfavorable conditions, they will change into gametocytes over here. Unfavorable condition. What is unfavorable condition over here? If the temperature is not good enough, if the patient is becoming anemic. Why anemic? Because they have already devoured. Here we are going to see only one RBCs over here, but the condition is not so. This can affect almost half of your RBCs also. And once the RBCs are broken out like this, definitely a patient will have anemic condition. And that is called unfavorable condition when the trophozoite changes into gametocytes. And these gametocytes are ready to be taken up by a feeding, blood-sucking Anopheles mosquito here. Now here you can see the gametes or the gametocyte is either a microgametocyte or a macrogametocyte. Macro is always a female, micro is always a male gametocyte. So this is still in the RBCs, and when the RBCs are taken by this mosquito, they change in the body of the mosquito. This microgametes or the macrogametes changes to form the proper gametes. Why don't they happen in the humans itself? Why don't they change into this proper germ cells in the human beings? Is because in the human beings, the RBCs are warm-blooded. The temperature is too high for the gametocytes to develop in the human beings itself. So that is the reason why they are in a dormant stage till then. And once a female Anopheles mosquito takes the blood, and once the gametocytes are in the body of the mosquito, the temperature comes down. The temperature of RBC is decreased. And once the temperature decreases, the gametocytes changes to proper gametes, and fusion of the gametes occur, whereby fertilization occurs, a zygote is formed. After a zygote is formed, a eukaryote is formed in the midgut walls. After the growth, they form into oocytes, and they are transferred outside the cells, oocytes. And once the oocytes completely grows in size, they rupture open to release the sporozoites, and all these sporozoites are ready in the proboscis of the mosquito. And this mosquito, when it is biting another human being, they infest all these sporozoites, and the life cycle occurs. So this is the life cycle of the Plasmodium falciparum. What are the other types of Plasmodium? We saw Plasmodium falciparum, Plasmodium ovale, Plasmodium malariae, and what is the other one?
Plasmodium.
Plasmodium?
Vivax.
Yeah, Vivax, Plasmodium vivax. Very good. So this is the advantage when you write down all these points in your rough book, so that it will be easy for you all to answer me. Fine. So these are the points. All this you have to go through it. I've already explained everything in this picture itself, so I'm not going to go into these points. Only thing what I'm going to say is what are the symptoms. You find extreme cold, chills, and fever. This is the symptoms first. Then you have dizziness, muscle pain, abdominal pain, nausea, vomiting, mild diarrhea sometimes, and dry cough. And in a very severe condition, you have high heart rate, high BP. That is, it can also lead to jaundice, enlarged liver. Why jaundice and enlarged liver is occurring in Plasmodium? Any idea? Why do we have jaundice during malaria? One simple reason. First thing is
Because they attack to the liver.
very much, very good. So they attack the liver, and what is happening?
Yes.
The functions of the liver is damaged, so they are unable
Yes,
to
sir.
produce bile, and that is the reason where we have this jaundice. The bile juice are
They also spoil the RBC.
exactly that leads to
That leads to
Yeah.
jaundice and enlarged liver.
yeah. RBC's damage leads to anemia. Liver damage leads to jaundice and enlarged liver.
Enlarged
Okay.
liver.
Yeah, that's good. So this was some of the important points on the plasmodium. Now coming to the next one, trypanosoma gambians or trypanosoma brucei. So this is also called as what sickness? Sleeping sickness. Okay, so they belong to phylum Protozoa, superclass Mastigophora. Again, we are going back to Mastigophora. What do you mean by Mastigophora? Anyone? Mastigophora. Mastig means
Flesh.
sarco. That
No.
is
Yeah,
No,
yes.
that's
sarcoidinous fleshy.
Yeah.
Whip-like structure.
Exactly, whip-like, whip. That is a flagella. Can you see that flagella over here? So this is the whip-like
Yes.
structure. So that is why they are under class superclass Mastigophora and class Zoo Mastigophora. Zoo means they belong to animal. We also talked about Phytomastigophora. Under Phytomastigophora, what was the example we did? What is the example of Phytomastigophora? Phyto means plant. I told you one particular one that is kept in both plants and animal kingdom.
Euglena.
Exactly, very good. Euglena is the one that is that belongs to Phytomastigophora. But here we are going to talk
Yes.
about Zoomastigophora.
Mastigophora.
Genus, check this point, genus Trypanosoma brucei, and then the species is different types of species we'll be talking about. So when I'm talking about the genus Trypanosoma brucei is the one that I'm going to keep on talking. Species will differentiate. Gambians, rodents, there are different types we'll come across. So this is a vector-borne disease. Trypanosoma is a vector-borne disease caused by infection of protozoan parasite belonging to genus Trypanosoma brucei, transmitted to humans by a sissy fly. So this is the fly I'm talking about. This is a sissy fly, and earlier we were talking about sandfly, right? So that's the difference you will have to. Sandfly is always blood-sucking ones, and that is the reason why they infect over there. Okay, so here they are transmitted to humans by a sissy fly bites, which have acquired the infection from human beings and sometimes from the animals harboring those parasites. There are two important subspecies you'll have to talk about. That is Trypanosoma brucei gambians. Trypanosoma brucei gambians. So this is the genus. Trypanosoma brucei is a genus. This is the species. They cause the African trypanosomiasis or West African sleeping sickness. Gambians leads to West African sleeping sickness, and rodensis causing the East African sleeping sickness. So rodensis is the East African, and Gambians is the West African sleeping sickness. And the third one is a brucei, a parasite usually attacking the cattle, and occasionally other animals are being affected, but you don't find them in humans. Now coming to the life cycle over here, when they are when a sissy fly takes the blood meal, they also inject the trypomastigotes inside the human beings, and once the injection of the trypanosoma inside the blood, what happens is this injected metacyclic trypomastigotes transforms into the bloodstreams, and they're carried to the other sites, and in the bloodstream they are multiplying in by binary fission, and they invade all the body fluids like the blood, the lymph, and the spinal fluid also. A very dangerous spinal fluid. Where do you find the spinal fluid? Any example of spinal fluids? Now we talked about blood, we talked about lymph. Fine. Where do you find the spinal? The spinal fluids, some of them in the cerebrospinal fluid. Yes. And your brain is lodged inside a water tank like structures. There's a water tank, and your brain is inside, and above that you have the skull. Fine. So in between that is the cerebrospinal fluid that acts as a cushion to your brain. Sometimes what happens is when you have an accident or when you hit yourself onto a hard wall or a hard structure, some freak accident occurs. What happens? The brain inside shakes, and when they are shaking, what happens is if they are going to get hit onto the skull is very calcareous, that is calcium carbonate, hard skull, they might get damaged. So to avoid the damage, your brain is encapsulated by cerebrospinal fluid, which acts as a cushion. There, just an example: put a ball inside the water, they are just floating, right? And nothing happens to the ball, it's just floating. So they are taking care of them. You jump inside the water, nothing happens. The same thing, if you jump on the ground, you're going to definitely break your bones. So your brain is safeguarded by the spinal fluids. And there are also some fluids that are present in the joints, the synovial fluid joints, yeah, between the two bones. For example, if you're going to talk about the knee bones, right, under that, you have those two bones, and in between you have those synovial fluid that is present, which acts as a cushion between the two joints, like that. Once the fluid is lost, what happens is the bones come together, and is when you have those severe pain. Arthritis, have you heard of this word, osteoarthritis or rheumatoid arthritis? Is when the fluid is decreasing. As we grow old, the fluid decreases, and once it is decreasing, the cartilage is lost over here, the outer covering of the bone cartilage is lost, and these two bone comes together, and when you walk, they come together, hit each other, is when we have that severe pain. That is how you find the old parents, they are unable to walk because of this pain. Okay, coming to our topic over here. This infects the body fluids over here. It can be a blood, the lymph, or the spinal fluid. Why we are talking about the cerebrospinal fluid is, if it attacks the cerebrospinal fluid, is when you have meningitis. Have you heard this word, meningitis?
No.
It's a very, very dangerous diseases of meningitis. The brain swells in size, and if untreated, then definitely the patient will die. So that is meningococcal or meningitis diseases of the brain. So you have to be very careful when you are dealing with this type of diseases. Okay, so what happens? The trypomastigotes multiply by binary fission in this particular fluids, different fluids, circulating mastigotes in the blood during acute phases, usually undetectable in the latent phase, but later on it becomes detectable, and they have two different types of phases in them: hemolytic stages and neurological stages is found in them. Before going into that, we will talk about what is happening after that. Some of the trypomastigotes are taken away by the CC fly in a blood meal, and then through inside the mosquitoes, they transform into vectors in the midgut, and then they become procyclic mastigotes, and from the promastigotes they become epimastigotes, and from epimastigotes they are ready to be injected into the another human being. So this is the life cycle. So what is happening in them? I'm not going to talk about this, I've already talked about that. I'm going to talk about two dangerous stages: the hemolymphatic stage and the neurological stages that we are going to talk about. Hemolymphatic phase, try to break that into two parts. Hemo is one part, lymphatic is one part. So when you're talking about hemo means
Blood.
very good.
Blood.
Yeah. And lymphatic is
Water.
lymph, the white part, the
lymph node.
WBC, the lymphatic system we are talking about. I told you the lymphatic system in the neck region, the arms, the knees. Yes.
region.
So the lymph region. So this you find in the blood and the lymph region. Incubation
Blood
period
cell.
is, yeah, RBCs. Incubation period is 1 to 3 weeks, and for that is for rhodesians, and for Gambians it is a little more longer. And the initial stage is known as hemolymphatic phase, which is characterized by non-specific generalized symptoms like fever, headache, joint pains, itching. This is also very important. You have a lot of itching sensation, weakness, fatigue, weight loss, lymphoadenopathy. What do you mean by lymphoadenopathy? Swelling of the lymph nodes is called lymphoadenopathy. And hepatosplenomegaly, hepato, liver, spleen, megaly,
Ma'am,
swelling.
what is fatigue?
Fatigue is tiredness, ma. You
Tiredness.
that is called fatigue. Tiredness.
Okay.
Right. So invasion of the circulatory and lymphatic system by the parasites associated with severe swelling of the lymph nodes. That is what is called lymphoadenopathy, swelling of the lymph nodes, often to tremendous sizes, become very, very thick ones. Shaista Mohammed, can you please mute your, yeah, thank you. The second phase is the neurological phase is the second phase. We were talking about the hemolymphatic phase, that's the first phase, and the second one is the neurological phase. This is a disease at a neurological level. Neurological means neurons. Neurons means brain, complete the neurons we are talking about.
Brain.
So in the brain, you call them as meningoencephalic stage. Meningoencephalic. Inside the brain, the skull, you have the skull. Inside the skull, you have that water, and in that water, you have the brain lodged in. So the water region around the brain is called that meningocele or meningoencephalic stage. Begins when the parasite invades the central nervous system. Your brain and the spinal cord is your central nervous system by passing through the blood-brain barrier. Sheikh Mastan, please mute your computer. Blood-brain barrier. What do you mean by a blood-brain barrier? Oh my God. Yeah. What do you mean by a blood-brain barrier? Is my time up? Yeah, this is the last one I'm going to talk about. Just give me 5 minutes. Blood-brain barrier.
Blood clot maybe.
Yes, ma'am?
Blood clot.
No. Don't get confused with the blood clot and the blood-brain barrier. Blood-brain barrier means the blood, the brain, and there is a the gates are closed. For example, if I have to show it in a diagram, let me put it out in a paper, kind of. So you have
blood vessels vascularized the central nervous system.
Yeah, this is the blood vessels, and you have the brain over here around. I'm just talking about only one single blood vessels. All the blood that is flowing inside the blood vessels will not come outside into the brain. What will come out into the brain is only the nutrients, the oxygen, and some amino acids that comes out into the brain, right? But sometimes what happens is this type of parasites will definitely come into the brain matrix surface, and this leads to different types of diseases. But mostly the blood that is present, the capillaries inside the brain and the blood that is present, the capillaries are having the tight junctions between the cells. Between the cells like this, they have those tight junction, which does not allow any passage of from inside to outside or from outside to inside. But here, this particular parasite can invade through the blood-brain barrier. Understood this point now? What is blood-brain barrier? Scientifically also, there is a lot of research going on over here to study a lot about this blood-brain barrier. Why? Because if there is any infection in the blood-brain, when you're taking some medicines, the through the capillaries, the drug is not able to go out and reach the target cells. So what they're doing is they have come up with this new technology called nanotechnology. Have you heard of this nanotechnology? Nano means 10 to the power of -9, right? So your medicines are going to become that small, very, very minute, -9, 10 to the power of -9, right? And they're going to prepare some drugs like that, which is going to be very small in size. They can go, move against the blood-brain barrier, reach the target in the brain, and they can give you some relief from all this type of diseases. So progression to the neurological phase occurs after an estimated 21 to 60 days in case of Rhodesians and 300 to 500 days in Gambians. What is the features of this particular parasite? Sleep wake disturbance. You're not having proper sleep, you're not able to wake up properly, you are always in a drowsy situation, like you don't know what's happening around the world. So that is called a sleep wake disturbance, leading to neurological stages of different diseases. That is what it is commonly called as African sleeping sickness. Okay. And what are the other symptoms of here? Tremor is shaking. Your body is always shaking. General muscle weakness, because the neurons are not going to give proper conduction of information to the muscle. So usually the muscle becomes weak. Hemiparesis, half paralysis. There are also paralysis of the limb, abnormal muscle tones, speech disturbance, gait disturbance. Gait is you're not able to stand straight. That is what is called as gait disturbance. Aesthesia, hyperesthesia, and anesthesia. Aesthesia and hyperesthesia is unusual itching onto the skin. Your skin is always itching. You are not properly feeling nice about it. And anesthesia is, you know what is anesthesia. Why do we give anesthesia? So that the patient will go to sleep and the doctor can properly do his surgery. So that is anesthesia type of disturbance in this particular disease. Visual disturbance, abnormal reflexes, seizures. What do you mean by seizures? Seizures are something but fits.
Convulsions.
Convulsions and fits, and sometimes leads to coma. There are also something like Parkinson-like movements. What is Parkinson-like movements? It's always a tremor that you find. Sometimes you find some patients, they are unable to stand straight. Their head is constantly shaking always. Have you seen any person having that disease? Constantly shaking?
Heroine Jamuna.
Exactly. Exactly. Heroine Jamuna has this problem. So they are constantly shaking their head. That is because of the Parkinson. Her Parkinson is not because of sleeping sickness. Her Parkinson is because of the neurological disease that comes into a different one. But here, the movement is something like that. That is what we are going to talk about over here. And of course, the speech disorders. So these are some of the diseases that you find in protozoans. And I hope you have understood this particular topic over here, protozoans. And I'm sure if you're going to get any question on this, you will be able to answer all these particular questions. Yes. Any doubts in this particular topic, Ma?
No, ma'am, you are explaining very well.
Ma'am.
Yes, Ma. Yes, Ma.
Ma'am, I have the book which I got from stationery.
Right.
So in this book there is test papers, ma'am. Can we prepare from that?
Test papers, you can take the questions, right? Answers, you read it from some other books, because sometimes they might give you wrong answers in them. These are something like model books, right? Model paper, something like that.
Yes, ma'am.
Yeah. Take some of the, you can read the questions from them, but the answers, try to read it from better textbooks. Clear?
Okay, ma'am.
Yeah. Any more doubts, Ma?
Ma'am, can you give us your phone number?
Take down 9490.
Yes, ma'am.
9490937422.
Thank you, ma'am.
Yeah, if you have any questions,
Thank you, ma'am.
Yeah, if you have any questions, do WhatsApp me. Don't call me because I might be busy. WhatsApp,
Ma'am,
I'll be
once
able
again
to.
tell your number.
9490 937422. So if you can share, what you can do is you can share my number in your WhatsApp group so that I can keep sending the PowerPoint presentation as and when we are completing the topics.
Thank you, ma'am.
Yeah.
Thank you, ma'am.
Or one of
Thank
y'all
you
can
so
just WhatsApp me and I will send the PowerPoint presentation. You can send it to your friends.
Ma'am, can you
Okay, ma'am.
give some important question?
Thank you, ma'am.
Again, important question as such, I can't tell you exactly because as and when I'm teaching you all, I will tell you what are the important points and important questions. You can just prepare accordingly. You just make a note in your rough book, some important question. Keep writing all those important questions whenever I will say. Okay, because you can't predict examiner what type of question he gives. We won't know. So better we be prepared for all the questions and we can be able to answer anything they ask. Yeah.
Okay, ma'am. Thanks, ma'am.
Okay, ma'am.
Thank you. See you all.
Thank you, ma'am.
The exams, ma'am.
Ma'am, nice teaching.
Thank you. Are we going to have a class on Friday, next Friday, or it's only on Saturday and Sunday?
No information, ma'am.
It's on Saturday and Sunday.
Friday, ma'am.
Because we were supposed to
Madam,
have a
please
class
give
on
me
Friday.
test papers, madam.
I don't have those test papers with me here. Again and again, I keep telling you all. You can ask Kishore sir, right? Do you all know who Kishore sir is? He's a counselor in Ambedkar University. He's the one who's coordinating all the classes. No?
No, ma'am.
No, ma'am.
Okay. You just find out from the office. Just call up the office of zoology department, take their number. You can ask all those doubts from them. Yeah. Yes,
Okay,
sir?
ma'am. Thank you, ma'am. Thank you very much.
Okay, ma'am. Bye.
Bye.
Thank you, ma'am.
Bye, ma'am.
Bye.